Hello, Miss Jody coming to you for our first video for biology. And so I'm just going to explain a little bit about what I want to try to accomplish during our time when we're separated. First of all, I want to try to keep the journals going. We know that we've already spent a lot of time putting these together, and even though we can't be together, it will be nice to continue our work. So I've come up with an idea that while we're separated, maybe we could work on getting a lot of our notes done, since that takes us a lot of time, and there's so much information in these chapters. So that way when we come back together, we can do a lot of our experiments and activities that go in the journal. So I do want to keep that going, and so I thought that would be the best way, because it'll be a little hard to do the experiments at home or some of the things that we want for the journal. And we can do that when we come back together. And hopefully this won't last that long and we can be back together soon. So when you're taking the notes, what I thought would be best is if you just take it on a half sheet of paper, because then when we get back together, we can glue them into the spots where they belong. Like, say for instance this, you see how this would fit right over that spot there, and so you could take the notes. Now you won't have the labels, but what you could do is just write on the back the title of the label of that note sheet, because I'll still have all the labels put into the journals, and we'll just glue the sheets right in order. And if you have it written on the back, then I'll know what note sheet that goes with when we start putting them back in. Also, I will be sending you in PDF form any of the visuals that we're going to work on, and you can print them out. I'm trying to be very selective and only doing like one page so it doesn't waste your ink. And so you could print that out, and then you can work on the diagrams slowly with the video, and then that way you can color them and make them a little nice. Sometimes we rush through those just because we don't have a lot of time in class. So that will be helpful, because then the diagrams would already be done also. If you don't have a printer, you could always just draw out some of the diagram on a piece of paper, and since you have a lot of time, you could sit there and work on that, and it wouldn't be as hard as if we were trying to draw it out in class. So that could help. There's some of them you won't really need to print, like say for instance this one that's going to go along with the experiment that I want us to accomplish. I did send this one because this might be something you can actually do at home. Normally though, if there's something that's going to be an experiment here, I would just print it out and I would put it in the journal. But this one you might be able to. I'll talk about that later, to have some experimenting with that. It's pretty simple ideas with plant responses. All right, so hopefully this will work out using the pieces of half sheets and keeping them organized for our return. Now, we just finished chapter 14 before we left. And as a review, I'll just flip through what we talked about. And let me move back here a little bit, these experiments. So let's see, where does it start? Okay, so it's when we begin to work with Kingdom Plantae. So as you remember, it took us most of the year just to get through the concepts of biology, what it means to be living, and all the processes and how the cells work, and just on and on, genetics and all that in-depth biology stuff. Pretty fascinating stuff, but it took us a long time. And now we finally have gotten to the point of, okay, now that we know how it works, then now we've got to take all the living world and start getting it in its categories, start organizing this giant living world. And so that brought us to classification, where we started classifying, trying to create the big categories, so the big domains, and then the big kingdoms, and then from the kingdoms down into the phylum and all that. So we worked on classification and then got the system ready so we know how it worked. And then now it's time to start taking all these living things and getting them in those little categories with classification system. So we worked on the smallest things all the way up to where we're at with Kingdom Plantae now. So we've already been through the living little guys, the archaea, the bacteria, and then even the non-living, like the viruses and the viroids and the prions, the agents, as we call them, since they don't meet the characteristics of life and at this point. And then we went on to what used to be called Protista and the splitting of that into Chromista and Protozoa, and then on to fungi and spent some time on fungi, and then finally we're on to Kingdom Plantae. And so Kingdom Plantae, we did the characteristics of a plant, classifying the plants in categories, like where do we start putting them in their boxes, and we figured out, okay, well, it can be about whether they're vascular or they're non-vascular. Do they have tubes? Do they bring the water up? Do they bring the nutrients down and around? Or do they just absorb like some plants that are non-vascular? We talked about using categories like, do they produce seeds? How's their reproduction? Do they produce, is it more about the spores, even though there's a generational thing there? If they do produce the seeds, do they produce a flower and they seal up their fruit, or do they have a cone and it exposes the seed? So there was a lot of categories to start with Kingdom Plantae. And of course with that we also went over all of the different terms that we have to understand in the plant world, like the different types of tissues, and then the tubes like we talked about before, the xylem and the phloem, and then all the parts of the plants, the nodes, the internodes, the leaves, the buds, all that good stuff. And that continued on in the structure of the plants, and of course all the real in-depth different types of cells and how they create different parts of the plants. And then we talked about the different categories like the monocots or the dicots, and those really being about the cotyledons in the seeds, but you could tell whether something was a monocot or a dicot with the parallel venation or the netted venation. So looking at the leaves and finding patterns to help us understand the different types of leaves can help us understand what kind of plant it is. So continuing with the structure of the plants, the leaves, the blades, the veins, stipules, all of that stuff. And then the different types of leaves, the evergreens, deciduous, on and on and on. Well, deciduous main. And then with the leaves, we looked at the structure of the leaves and how the gases are exchanged coming in and out on the stomata. And then we collected some different types of venation and was looking for patterns in the leaves. And here's my little parallel venation with my monocot there. And then we talked about this coming in and out. This was a flat version, and this is the side version of that, seeing this openings, the mouths of the leaves that allow the gases to go in and out, and how that's structured. Then we also, obviously I'm missing my pictures in my book, but we'll add those later. We talked about how the stems work and the growth, and this is supposed to be my tree roots, not roots, the trunk of the tree and how it grows out. And so we did that and then labeled the bark and what was part of the bark and what was the vascular cambrian and what was part of the wood. You remember that. We did that on the side. I haven't drawn my arrows, but you get the point, and we collected some of that bark. And then we went on to the stems and the roots note page. So we talked about the different types of stems and the main two groups, like the parts of the stem, but then also the main two groups, herbaceous or the woody stems, and sort of how they form and what's going on with them. And then the roots, we spent a little time with the roots and whether the root system would be tap roots or fibrous roots and how the roots work. Then we went on to the gymnosperms, and that was the ones who don't necessarily produce that fruit, or they don't seal up their seed in a fruit. Instead, it's a cone. And so we talked about the pollen cones and the seed cones and then how that works for fertilization. And this was our little doozy. Somebody did this one for me. Can't remember who I asked to do that for me, but we rolled the the pine cones, and my color choices were a little off here. We're gonna pretend like I meant to be very abstract and just wild here. And so we rolled the cones, which is representing the cone bearers, and then of course created an evergreen. This was somebody's evergreen they made for me. Little cutie there. All right, and then we went on to the angiosperms, and I know there was way more notes than this, but it looks like I did not take all my notes in my book. I sometimes do that, and I can't do that now because I have to have them ready for you to see, so you can use those to create your notes. But we've already done these pages, so no worries there. So we talked about the angiosperms, that's the flowering and fruit bearers and such, and them being such a big group, 80% of all the plant species, and pollination and how that worked in that process with the flower there. That was way more exciting, so 3 real notes on it. This was our flower dissection when we started to work on getting the petals off and counting them, and that will help us understand the petal arrangements and the amount, along with some other things like the leaves and all that, can help us with classifying plants. And so that's something that we had there. We'll have to make sure we get back to seeing what those numbers mean. Then we had the seed world, and we just finished this up. This was kind of where we ended, understanding what the cotyledon was, and that is the, if you remember, when you have your seed versus your spore. So they both, the seed and the spore, both have an outer coating. They both have the baby plant that wants to come out, but the difference between the seed is the seed also has this cotyledon, this extra food source, where the spore doesn't have that. And so if the spore lands in not favorable conditions and doesn't have the ability around him to get the nutrients, then he won't make it. But the seed can make it for a while without a great nutrient source. And so when the seed does germinate, doesn't even need to photosynthesize. Once the plant pops out, we don't even have to start photosynthesizing yet, because we have a little bit of time because it's eaten off of the feeding off the cotyledon. And then, of course, the cotyledon, if you have one cotyledon, you're a monocot, and then if you have 2 cotyledons, you're a dicot. And so we learned a little bit about germination and what was necessary to break that seed open. And so you had to have a temperature situation, the oxygen, because you still need oxygen for cellular respiration to happen in the cells, and then also moisture, so that the seed coat will swell and break it out. And that was one of the experiments that we worked on, and I have our experiments from the last time we were together. I'm going to just take this off here and show you. We were trying to germinate the seeds, and this is, I guess they didn't name there, but maybe because it was already a label on there from one of my other classes, butterflies. So the butterflies, that table, those little cuties, your jar did not break. I don't know if you didn't put enough water, is my guess. It's bulging at the top, but yours did not break. The other team that did and was successful, it was called Beware the Bean, Church of the Bean. Bean, 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 bean. Okay, so that table, the Church of the Bean, you were successful at breaking the jar. As the seeds germinated, the pressure inside, being trapped inside, was so strong as the plants, as the babies started to try to come out and the coats swelled. So you were successful, and I think it was like the next day yours broke. I put it in these containers so it wouldn't shatter. This class, which was, afraid to touch this one, it's bulging so much. I'm going to cut my hands here. Austin, Shelby, and Johnny Z. Yours did not break yet as of now. You might not have had enough water, and so you had to make sure it was really packed with water to get that to swell open all the seeds and allow for that germination. But yours might still look that nice bulge there. These are a smelly mess, I will tell you, and I'm not going to have fun doing a video with them right here, but I had to bring them over for you to see. All right, so I'm going to put this back in my little homemade contraption here for our video. And so that was the end of that particular unit. And we will put some pictures in there of the seed germination project. Oh no, I guess there was one more. The fruits. Remember we did the apple stamping and we talked about the seeds that are actually sealed up for protection inside of a fruit. I've got to draw in, I've got to find a little Sharpie here, and I'll draw in my little seeds with the Sharpie and then my stem and all that. You did some apple stamping, and I put those in there in our books also. And put some seeds on those little doodaddies. And then we talked about the fruits, classifying the fruits. And so these fruits, which are sealing up the seeds, there's different types. There's dry, of the simple fruits, there's the dry fruits and the, which I always say fleshly. Wait, no. Freshly. No, fleshy. There we go. Fleshy fruits. And so the categories of dry type of fruit and versus the flesh type fruit that we're familiar with. And this is also in your book, so you can remember that. And then of course the compound fruit, those who come from multiple of the ovaries, if you remember that weird concept. And so that gave us our little diagram there. And then of course we have a picture page, and then we were supposed to have quiz 14, which we were supposed to do when we returned, and then everything happened and we haven't been able to return. So I don't know if I'm gonna eventually just put that in a PDF and send it to you, or if we're gonna just, hopefully, if we're not gonna be out too long, we can come back, and when we come back doing all of our activities and stuff, we can take some of the tests. And I don't think it'd be that big of a deal to take some of it together, because we're gonna have some learning challenges with this new format, right? So we'll see. But anyways, so now we start chapter 15, and this is continuing on Kingdom Plantae. It's just now going to be focusing on the side of the processes, the things that are going on within the plant. And we've already learned a lot of this because we had to learn photosynthesis and all that in those biological processes early in the year. So that's good. Get that out of the way. So now it's just more of the application side of it a little bit. Other little things that aren't as in-depth as the photosynthesis and stuff like that. So that'll be good. But so as we look at this chapter, we'll be looking at specific things that happen in the plant world. Like one, transporting the nutrients in the water. How does that really work? We already know, because we already talked about vascular tubes. We already know that any plant that has tubes, it's there's two of them, two sides to it. There's the xylem and the phloem, and the xylem's bringing the water up. Remember I talked about using, like, thinking of ding, 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 a xylophone to remember. It's bringing it up, the water and the dissolved nutrients, all the minerals and stuff that the plant needs for life sustainment. And then the phloem are the set of tubes that are going to bring all of the sugars that are produced in this process with photosynthesis and to the leaves for storage, and then also for growth as areas have to continue growing. So we have that system, and that one, if I put some arrows here, it can go up and down, and sort of, I guess I put side to side. It usually says up and down, but really it seems like it would go side to side within the different stems and all that. So that normally is illustrated up with the xylem, down with the phloem, but really it's kind of, the phloem has got to go a little bit everywhere to kind of give everybody what is needed. Now, these tube things, that's a simple version of it, but the question is, like, how does that really happen? Because we know how we get our water, how we bring it in with our hands, and we drink our water and then it goes through its system, and we know how we bring in our food, and we've got our esophagus and our stomach and our digestive system that's all, you know, doing what it does to break everything down, and then all the nutrient absorbents. But plants, you know, they don't have the hands to pick up a glass of water and drink, and they don't really go to the bathroom like that. So, like, there's a lot of different processes that goes along with the plant world. So we have to figure those out. And so the vascular tubes that are moving things up and down, we really want to understand how they work. So one thing to note is all of the fluids and liquids that go along with the xylem and the phloem, like the water, the nutrients, the sugars, everything that's moving around in the plant is what we would refer to as sap. Sometimes you think of sap as like just that sticky stuff, and it is because it's got that, obviously, that sugar base, or that sugar in it, and many times. But the sap is going to be a term that's going to say, okay, all fluids from the phloem and the xylem. Now you might say, okay, the xylem sap or the phloem sap, because you sap, but you still want to separate them. But it's the sap world, just so you know that. Okay, so let's look at the xylem, how that is thought to work, and then the phloem. Okay, so with the xylem, it's primarily carrying water and dissolved nutrients up, right? So how is that going to be functioning? Well, there's theories behind how these things are working, and obviously we try to study plants, and we have a ton of different methods to study them. But again, just like any model, things can there can be ideas about them, and they might eventually change, you know, as we get more information. But the idea of how the xylem is working is pretty simple. First, they're saying, okay, the roots absorb the water and the dissolved minerals from the soil. So you got all the water and the dissolved minerals, and the roots begin to absorb them up, right? The root cells that are in the roots, they use active transport. Now you remember active transport when we're talking about cellular processes. The active transport, you're going to have what? Some energy. Passive transport, you don't need the energy, but active, you're going to be having some energy to push that through. So root cells use active transport to increase the amount of dissolved minerals as it's coming in, so that osmotic pressure moves water from the soil into the root cortex. So what that's saying is the cells begin to take in like a whole bunch, through active transport, a whole bunch of these dissolved minerals, so that then in the end, one side, just like when we had the osmosis egg, one side has too much. And so what will the water always do? It'll try to even it out. And so in the osmotic pressure, we see that the side that has too much of the dissolved stuff in it, the water is going to be moving in from the soil into even it out. And so that's going to help move that water up. And so incoming water pushes water already into the root deeper into the cortex. So it just keeps trying to even out, pushes it up, trying to even out, pushing it up. It keeps bringing the nutrients in and allowing for that process of movement. And so we begin to have an upward movement into the tubes. Okay, so then what happens after that? So the water is then filtered through the endodermis as it moves into the xylem. And I have like a little diagram over here so we can illustrate this. And this is the tip of the root and the root hairs and the phloem and the xylem. And you see in your book, there's a diagram showing you how this is working that you can work on to fill this in. I see it, pages in 286. And it will just give you a little information on that. And you can see it right down here, what's happening with the root cells, the soil particles. And so here's the little root cells bringing it in, and the xylem and the outside cells that are doing this, filling up with a whole bunch of stuff so the water will suck in and even it out. So you can work on that diagram, understanding this part of it right here. And so with that, I'm not going to do my colored pencils on this one because that one's pretty easy. I'll use the colored pencils to help with the second one because that one is a little bit more, that one will be more helpful to see exactly what's going on the phloem. So this one will represent the way the water moves up, and this one will represent the way the sugar and all of that moves throughout. Okay. So on your diagram, you would show how it's coming in. The root pressure causes the incoming water to come in. So you draw your little water and your soil around here and your water coming in through the roots, and then the osmotic pressure helping with that movement up. Okay, but even though it does all that and it starts moving up, it's not necessarily going to be enough. And there is what we call the adhesion side of things. And we've already learned the adhesion and cohesion. If you remember, it's back in chapter 2, maybe something like that. So if it's adhesion, it's that the molecules, a certain molecule is trying to hold on to a different type of molecule, not the same type of molecule. So that would be like water trying to hold on to the side of a straw. And so when it does that, the straw obviously is a different made of different molecules in the water. But when it's trying to hold on to it, it's able to adhere to it a little bit higher than the level of the water in the rest of the cup. And so it creates what we call capillary action, where it can, and especially with small tubes, you can get a lot more capillary action with small tubes. It'll start pulling it up, and because of that adhesion. So you have adhesion, so you got the bottom of the root, the water coming in through osmotic pressure, and it keeps pushing it up because of trying to even the solutes out. And then we've got the adhesion helping, and with the capillary action to pull it up. But then there has to be more, because we have giant plants that like giant trees that they tell in the book about the sequoia. And how do we get water all the way to the top? I mean, what else is happening here? Because you can do a little capillary action and stuff, but is that going to be enough to get it all the way to the top? And so they give the theory of the cohesion side of things. Now, if you remember what cohesion is, adhesion is when two different molecules, like the water's trying to hold onto the side of the straw. But cohesion is when water molecules want to hold onto other water molecules. They want to hold onto each other. The same type. And so the water molecules that are already in the xylem, there are other water molecules above it, like say from the areas of the leaves and other parts of the tissue. And so the idea is, as water moves out of a system, so as the water is moving up, okay, so let's say it's moving up, there's this process called transpiration, and you learned that back when you learned the water cycle when you were a little bit younger. And that's when, like evaporation, the water going up and then forming the clouds and raining back down, that part of evaporation in the cycle. Transpiration is when the leaves are releasing some of the water vapor and getting it back into the water cycle, like 95% of it actually, so it's a lot more than a little. And so in this transpiration process, it's moving out of the leaves. Well, when it moves out, then it's going to move from one area and pull the next water molecule with it and keep pulling it up. And it's almost like a tug. Just keep tugging and tugging and tugging and tugging and tugging in this process of cohesion. And so this is the theory, okay? So it comes in, it's got all this pressure, and osmosis is trying to even it out because of how concentrated the solutes are, and then also the pulling up, and of course the adhesion. So you got a lot of things going and try to get that water up. And good thing that works, right? And we hope that's exactly how it works, but that's the theories. Then we have the other side. Okay, so we can get the water up. How do we get all the stuff that's being made everywhere else without, this major pumping system that we would normally see in living organisms like us? How do we get our blood everywhere? Well, we have a heart, so there's a pump. So it's weird with the plant world, like, how's this doing everything? And again, we know a lot of this comes down to the cellular side in the amazing world of cells that's worth its own year of study in cytology. So when we look at the phloem, we see that the phloem is going to be able to go different ways. So it's not just down, but from where it needs to go to get to all of the parts to bring all the goodies. Okay, so the phloem is a primarily carrying the sugars from leaves to storage and other areas of growth. And there's my little doodads there. And I try to move this up a little bit so that you can see the edges. Oops, might have knocked my phone there. How's that? Probably shouldn't have touched it there. Now it's got to refocus in on there. Sorry about that. Okay, so with this side of the notes, and I tried to squeeze it in, so this is why it's a little harder to see. The phloem, we have some steps too. Now this isn't this 1, 2, 3, 4, 5, 6. This isn't like the 6 steps of the phloem or the 7 steps of the xylem. That's just Miss Jody trying to keep track of what's the next step. I don't know if there's a certain number to any of this. All right, so other than when we first get started, the cells that make the sugars called the sources are found in the leaves and some of the herbaceous stems. So I guess I do have to unfold this so I can slide this over and show you some of this too. So hopefully you can still see it. But So what we will have in this time of bringing the sugars and all that have been made all over the plant for nutrients and storage, we have what we will call the source cells. So this is what, this is going to illustrate the source cell. And I'll make that, because I think the book uses red. The source cell is the cells that are the ones that actually make this stuff, the sugar, through that process of photosynthesis. And so say you have this source cell, it's at the leaf, you can see. So the source cell is going to be making all these goodies and ready to kind of give it to whoever needs it, right? And so in this process, the source cells filled up. And then we have also what's called the sink cells. The sink cells, and just think of it as an empty sink, and with a little drain on it, is just ready to like take all of the nutrients. Now these sink cells, these are going to be cells that require large amounts of sugar. So things that are like growth areas and such that are going to need a whole bunch of sugar to keep doing what they're doing, those are going to be the sinks. And that's where a lot of that sugar is going to be going to. And so this diagram helps us understand how the source makes it and then how it gets down to where it needs to be, and in that process creates a pressure flow system. And so that's where this pressure flow hypothesis comes from. Well, you can use this in all physics, but it can be applied to the plants. And so it's kind of like they say in the book, it's like a garden hose. When you first turn on the garden hose, there's a whole bunch of pressure coming from the tap. But then at the end of the garden hose, it's just like slightly like flowing out, you know? And so the pressure is being created to make the water flow, and then it flows through at the end. And that's sort of what happens here with the phloem. And so we've got the source cells and the sink cells, and we will begin to create a pressure system to get everything to move, since we don't have a pump there. All right, so the source cells, they use active transport. So again, active transport to make sugars, to move sugars into the water-filled phloem. Okay, so if you look at this diagram, you've got the phloem on this side, which is the ones that are going to bring all the nutrients, and then you got the xylem over here. Now the xylem is the one we're talking about, water and nutrients that are constantly going up here. So they are, if you look at a cross-section of a plant, you'll see they're kind of mixed and right near each other. So a whole bunch of xylem and phloem are hand in hand. It's not like there's one tube of xylem going up to everything and one tube of phloem going up to everything. It's a cross-section of like tons and tons of these tubes in the stems and moving through. And I think we looked at that back when we studied the stems. I think they had a cross section. Let me see if I can find that. But do you remember, or I might have put one up, but I think, let's see, the roots there, no. Well, in the root tip, it kind of shows you this on this one. It's not the best because I'd like to see an actual cut of the stem, but you see where it says phloem here? The phloem are intermixed with the xylem. So they're near each other. And so that's my point, is that you got tubes going up, you got tubes going down, and they're all intermixed in a whole bunch of them, not just one. And so with these tubes, we've got this water that's always going to be moving up through the process we talked about a second ago, with the idea of being pushed up, and then the adhesion and capillary action, cohesion, all that. And then we got these guys who are making all this sugar and stuff. So the cell will use active transport to move these wonderful sugars into the phloem. And this is the area where the sugars are going to need to be moving through to get everywhere where they need to go, because those are the tubes that transport them. It doesn't move them into the xylem because the xylem's got its own job, but it moves them into here. But these guys are right next to each other, and so we're able to have a flow between the two. So water can flow into the phloem. And so with that being able to happen, like we see with any of the processes of cell and getting in and out of cellular membranes and all that, the water can go in here. And so this is a water-filled situation with the phloem. So the source cells use active transport to get it into this water filled phloem, and that will help with the transport. Now, the thing is, why would the water go in there? And it all goes back to osmosis, just like everything goes back to osmosis, right? This is why you can't take biology and not do the osmosis egg, and it never gets old. I don't know how many osmosis eggs I've done. It never gets old. It's still an exciting process, and hopefully you will forever be as corny as me and love osmosis eggs. So higher concentration of sugars causes the water to move osmotically into the phloem from the nearby xylem. So because we start getting all of these higher concentrations over here, then these guys that are next to each other, the water has to do what the water always has to do. It's got to even out. It's lots of concentration over here, so it's got to move over there and try to even it out. And when it does that, it starts this movement of pressure that's going to help us. And so with that note there, the higher concentration of sugar causes the water to move osmotically into the phloem from the nearby xylem. Okay, then at the same time, the Cinq cells remove the sugar by active transport, and then the water's diverted back. And so, of course, the sinks, like we said before, that's the areas of need, so they're going to be removing it and using it and all that good stuff. And so when that happens, then now we had some, lots and lots of concentration, so the water had to move in and even it out. And as it's doing that, then when some of those sugars and all that get here, and then this is moved out, then it's not as much concentration down here. So if you see, there's like 4 little dots compared to a whole bunch over here. So by the time some of this is moving into the sinks, then